Hello friends. In the previous classes we are discussing on the enzyme definition, enzyme characteristic and different types of the enzymes. That we continue with the enzyme, that's a nomenclature. So International Union of Biochemistry and Molecular Biology, they have given name for the systematic classification of the enzymes. So the first going to the nomenclature. So in the nomenclature, the name of the enzymes end with ASE. In the enzyme nomenclature, so usually all enzyme, proteases, so isomerases, so like that all enzymes are end with ASE. That's a one condition. Each enzyme name consists of two words. Okay, one is a enzyme. The first one denote the substrate and second indicate the reaction. There will be a small spelling mistake. The first word indicate the substrate and the second word indicate the reaction. For example, hexokinase, HEXOKINASE, hexokinase. Okay, here the two words will be there. AS is a separate one. Hexos means one word. So like that, the first denote the name of the substrate. Second one reaction is the name of the reaction. That's a hexokinase. Okay, so like that, if it is involved in the transferase, then it is transferases. And sometimes they are called as oxidoreductases. So like that, there will be a different types of name. Based on nomenclature, there are 6 classes or enzymes are mentioned. 1 digit, that's a major class. 2 digit, that's a subclass. 3 digit is a subclass. So like that, there will be a mention of the exam. For example, alcohol dehydrogenase, that's a 111 system of classification. So going to the major classifications, that's the first one is oxidoreductases. These are the 6 major classes. That's oxidoreductases, transferases, hydrolases, okay, so isomerases, liases and the ligases. So all these are the 6 classification of the enzymes. So in the exam they will ask, explain the classes of the enzymes. So usually for the 5 marks question they will ask this type of the question. So first one is oxidoreductases. So what is oxidoreductases? The enzymes which catalyze the oxidoreduction between the two substrate. Enzymes which catalyze oxidoreduction between the two substrate. Transferases, enzymes, next one class is transferases, enzymes catalyzing a transfer of the group between the two pair of substrate. That means here the enzyme catalyzing act like a transfer of the group between the two pairs of the substrate. Okay, then third one is hydrolases. The enzyme catalyzes hydrolysis process. The enzyme catalyzes the hydrolysis process. Those are referred as the hydrolases. The fourth one is liases. Enzymes that catalyze the removal of a group from the substrate. Okay. The next one is the isomerase. Catalyze isomerization reaction. Those are called as the isomerases. Sixth one is the ligases. The catalyzing the link together of the two compound. So each one have there will be a specific action or the reaction in the nature. So based on that they categorized into a 6 type. First one is oxidoreductases. There will be a oxidation and the reduction between the two substrate. Next there will be a transfer of the group of the substrate. The third one is hydrolysis activity which takes place. In the liases, that's a removal of the substrates. Isomerases is involved in the isomerization reaction. Ligases will be involved in the linking together of the two compounds. So like that there will be a different group. For example, oxidoreductases is example is NAD, oxidoreductase cytochrome C. So these enzymes are the oxidoreductases. Transferases, that's example, they will transfer the acyl, alkyl, amino, phosphatase groups, etc. And glycolate, that's an example, or phosphofructokinase is an example for that group. The next one is hydrolysis. That's example for the hydrolysis, sucrase, maltase, fructose. So those are the hydrolysis. So next one example for the lyases is the octinoses, aldoses, PEP carboxylase. These are the example for the lyases. The example for the isomerases is the phosphofructoisomerases. This is an example for isomerases. Example for the ligases is the glutamic synthase. Okay, so like that there will be how the major 6 classes of the classification of the enzymes involved. Next, going to the nature of enzyme action. Each enzyme have the an active site into which the substrate will be bind. That means each enzyme have an activation site. So usually the substrate will bind to that active site. The substrate bind to the active site of the enzyme fitting into an active site. The binding site of the substrate induces enzyme to alter its shapes. The next is the activate site of the enzyme is now closely proxy of the substances break the chemical bond of the substrate into a new enzyme. That's a product complex will be formed and releases the product into the reaction. So that's some of the enzymes that will be involved in a enzyme inhibition. Some of them will be promote and some of them will be inhibit. So the inhibiting enzymes are the competitive, non-competitive and the feedback enzymes. Okay, so going to the factor affecting the enzyme of reaction. What are the factors? Temperature, pH, substrate concentration, enzyme concentration, and also the concentration of the enzyme product that will be affected to the enzyme of reaction. So I will go back to that. Enzyme inhibition. How actually the enzyme involved in the inhibition by competitive mode and also the non-competitive mode. Just like so, when the reaction will takes place, they will bind to the substrate. They may increase the reaction or inhibit the reaction, and there will be a enzyme involved in a more promotion. That's why they called as a biocatalyst. But sometimes they may be inhibit also. They are called as a competitive and the non-competitive enzymes. So next, going to the factor affecting, already told, that's the temperature, pH, substrate. So all those factors will be key role in the enzyme reaction. So that we will study in the next class.